Welcome, everyone. Thanks so much for joining our webinar today on a new era in responsibility, the NIST Gut Microbiome Reference Material. My name is Gwendolyn Jones. I have the privilege and honor of serving as the Executive Director of IFINS, and I want to also call out my colleague, Marie Latulippe, who serves as the Director of Science Programs at IFINS. And as we get going here today, I want to take a moment and orient everyone to who IFINS, or the Institute for the Advancement of Food and Nutrition Sciences, is. We are a nonprofit organization grounded on our 4 core values: scientific integrity, centered on transparency, collaboration, and public benefit. As an organization, IFINS mobilizes our members from government, industry, and academia to drive, fund, and lead actionable science, focusing on food safety and nutritional science to advance public health. We catalyze the creation of scientific knowledge by funding external researchers and convening experts to support positive change. Our membership is broad and diverse, and it is our collaborative and inclusive structure that empowers our members to bring forward the different perspectives across the entire food and beverage ecosystem. As we get started, since folks often ask, how is it that you guys, dare I say, do what you do? How is it that you catalyze that science that matters? I want to highlight that as an organization, we work largely through our member-led committees. We have 14 of them total, ranging from our Assembly on Scientific Integrity to our work on protein, right? From our work on carbohydrates to food microbiology. Each one of these committees, very similar to the IFINS Board of Trustees, is comprised of members from government, industry, and academia. So with that, one of our committees is our Nutrition for Health Committee. And this committee is focused on advancing and communicating the scientific understanding of the impact of diet and dietary constituents on gut and host health. And today we're going to learn more about some work that we've got going on, which is a new study on advancing research that supports gut health, specifically looking to document the continuum of measures from health to disease. And we hope everyone can join us at Nutrition 2025 as that new project is explored further. As we get going here today, since different folks have varying levels of familiarity with our GoToWebinar platform, we do want to highlight that we do welcome your questions. We welcome your comments as well. On the right-hand side of your screen, most likely on that interface, what you'll see is that question box. Please do feel free to type in your comments and questions there. Comments and questions are only visible to IFINS staff, right? And at the end of our webinar, those comments and questions will be brought forward to our presenters. So speaking of presenters, I want to take a moment and briefly introduce our 3 presenters today who are going to talk to us about this new era in reproducibility, the NIST Gut Microbiome Reference Material work. So joining us today will be Dr. Scott Jackson, who's the group leader of the Complex Microbial Systems group at NIST. We have Dr. Sandra De Silva, who is a research chemist in the biomaterials group at NIST. And will also be joined by Dr. Stephanie Cervantes, who is a microbiologist in the Complex Microbial Systems group at NIST. So with that, let me turn the presentation over to Dr. Jackson, who's going to walk us through and share with us how we can all be a little bit more reproducible. So, Dr. Jackson.
Thank you, Gwendolyn. So I need access to share my screen. All right, good. So thanks, Gwendolyn, for the very nice introduction, and to your colleague, Marie. who's been our partner on this for many years, and we're really excited to share this output that has taken us many years to arrive at. As Gwendolyn said, I'm the leader of the microbiology group here at NIST. Our program focuses on measurement science around the clinical applications of the microbiome. I'm going to delve into this a little bit more. Why don't I just go ahead and start? Standards to support industrial innovation in microbiome science, which includes food science. So let me take a second to introduce NIST. I feel obligated to introduce NIST when I speak about NIST outside of NIST. So NIST, of course, is the National Institute of Standards and Technology, which is a non-regulatory federal government agency within the Department of Commerce. So just some fun facts about NIST. We're the oldest technical institute in the U.S. government, dating back to 1901. We have two large campuses, one located in Gaithersburg, Maryland, and another in Boulder, Colorado. Here are pictures of those two campuses, and I bet if you look closely, you can tell the difference between which one is in Gaithersburg and Boulder. About 5,000 scientists and engineers strong, about $1 billion annual budget. And owing to our roots in the Department of Commerce, our mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science standards and technology. So I spent the first half of my career working at the Food and Drug Administration, the FDA, which is a public health organization. And it took me a while to sort of reprogram my brain when I jumped from FDA to NIST to say, okay, I'm not working in a public health agency anymore. I'm working in the Department of Commerce. And we have to sort of reframe how we approach science and how we approach impact and stakeholders. Okay, here's another fun fact about NIST that I like to show. According to several independent studies, every dollar invested in NIST has returned $9 to the U.S. economy, and that was published in Science a few years ago. So something to make you feel good about government service in the current climate. So we think a lot about the bioeconomy, and moving on, perhaps the thing that NIST is known for, most well known for, is the production of reference materials. It is by no means all of what NIST does. It is a very small part of our NIST portfolio, but nevertheless, it is probably the thing that we're most well known for. As you can see in this display cabinet, which is in our headquarters building, you can see we have standard reference cigarettes. We've got peanut butter and meat homogenate. Here we have some industrial sludge, domestic sludge, Portland cement, etc. Some of these might seem silly, like why does the U.S. government need standardized cigarettes and peanut butter? But I promise you, every one of these standards was developed for a very important and practical industrial application. Developing a reference material is not something we commit lightly to. It typically takes a small team of PhDs about 5 years to produce any one of these. So, it's not something we just take lightly. We put a lot of thought and do a lot of stakeholder outreach before we commit to the development of any reference material. And every one of these reference materials has a long and interesting story behind it that some scientist could tell you. I just want to point out quickly that Sandra, by the way, you are unmuted, and I'm hearing some background noise that might be coming from you, so just FYI. So a lot of the work we do in the biosciences space here at NIST falls under the regulatory purview of the FDA. So we do a lot of standards for diagnostic testing and for biomanufacturing quality control. So this slide sort of highlights our relationship with Food and Drug Administration, our relationship with industry, which all centers around industry standards. So NIST engages industry, we engage FDA to understand what standards are needed. We then build and deploy those standards, and it works in a very harmonious, productive way. The FDA's regulatory decision making is benefited from the availability of NIST standards. Okay, so why microbiome? Probably no surprise to you all. The human microbiome has emerged as a new target for diagnostics and therapeutics, and industry has responded accordingly. In the last 10 years, we have witnessed the an entirely new bio industry that has emerged around the human microbiome space. I attended a conference a couple of years ago, and I stole this advertisement from the conference website, which just showed some of the companies and academic institutions and government agencies that were part of this microbiome conference. And you can see there are big pharma companies there. I see Pfizer up top. You can the smaller ones that you may not have heard of, or smaller startups that are working on some technological discovery, maybe from an academic lab that they're trying to move to the clinic. You can see big universities on there, government agencies. So again, this is the bioeconomy. We're in the Department of Commerce. This is why we do what we do. This isn't just wishful thinking. There have been some real progress. So perhaps the biggest news that we've seen in the last few years is that the FDA approved a couple new microbiome therapeutics. This is one of them, Reproducibility. If you could probably assess by looking at that picture on the right of what this material looks like. It is a fecal-derived therapeutic. So this is a drug according to the FDA's definition of what a drug is, and it is manufactured from human fecal material. It's been shown to be highly effective at treating recurring Clostridium difficile. This wasn't just a how do I get that to go away? Okay, well, it's going to be there. So this wasn't just a one-hit wonder, but about a year after the approval of Reproducibility, we saw the second FDA-approved microbiome therapeutic, this one from the pharma company Seres, based out of Boston. This is Voust. Again, importantly, this is manufactured from human fecal material. So this is a fecal-derived FDA-approved drug, right? So here's real progress being made in the microbiome clinical translation of microbiome science. And one last thing I want to point to, because the oncology community has really blown up in the last 10 years, leveraging the gut microbiome and its immunomodulatory impact on cancer and cancer therapies. So in 2018, there were 3 back-to-back papers published in Science that all reported the same finding, albeit independently, and that is the gut microbiome modulates the efficacy and response to immunotherapies in cancer patients. So these PD-1 checkpoint inhibitors that are being used to treat cancer, and by the way, these PD-1 checkpoint inhibitors won the Nobel Prize, I think, in 2020, maybe. The efficacy of these checkpoint inhibitors, these life-saving drugs, immunotherapies, is modulated by the gut microbiome. And you can believe that oncologists today are now trying to leverage the patient's gut microbiome by doing fecal microbiome transplants and microbiome profiling to make these cancer therapies more effective. And they've made incredible progress over the last 10 years. So let's talk about measurement assurance and the need for validation and standards, et cetera. So whether you're manufacturing a probiotic, or whether you're manufacturing a fecal microbiome transplant, or whether you're manufacturing some new microbial therapy, you have to demonstrate the quality and safety of that material through the development of some analytical methods. And these analytical methods will typically ask certain questions about the purity, potency, identity, stability of these materials. So let's just think, well, what are some of the questions that need to be asked of these microbial products? First and foremost, well, what microorganisms are present in the material? And we might want species or strain level designations. What are the relative quantities of these different microorganisms? So in a complex microbiome, it's not just a 1-to-1 ratio of all of the species that are there, but rather they're there at differing abundances, and this has some ecological fundamental reasoning behind it. So to understand what the relative quantities of the different microorganisms are is an important quality attribute of a microbial product. Are the organisms viable? Very important question. Are there any contaminating organisms or pathogens? If you biomanufacture anything that's going in or on the human body, you have to demonstrate that it's free from contaminating microbial organisms. But what if your product is a big concoction of microbial organisms? Now that question becomes a lot more challenging, because it's not is my product contaminated with microbes? It's what microbes are supposed to be there and what microbes are not supposed to be there. How stable is the material? What's the potency of the material? Look at this fecal microbiome derived material. Like, how do you do a potency assay on this? That's something that the FDA requires. What's a dose? Thinking about batch to batch variations from one manufactured batch of material to another. This is especially challenging when your material is fecal derived, because probably no surprise to any of you, people don't poop the same material every day. I mentioned these just to mention that measuring these attributes requires validated analytical techniques. You need reference materials, benchmarking studies, validation studies, measurement innovation, etc. And this is what NIST does. Okay, so moving right along, what we advertised this webinar for is to highlight our new fecal standard that we released about a month ago. Here are some pictures of NIST scientists in various stages of manufacturing large quantities of human fecal material. As you can see in that center picture with the scientist standing in front of this Ninja blender, homogenization of the material is very important. Poop is inherently heterogeneous, and for a reference material, you need a lot of homogenized material. So we spent a lot of time doing R&D to figure out how to manufacture this material in a way that makes it homogeneous and stable. So we decided early on, for various technical reasons and practical reasons, that we wanted, pardon the pun, but 2 flavors of fecal material. And we decided that the best way to get these 2 different types or flavors of fecal material was to recruit 2 different donor cohorts that differed by some significant attribute. And knowing the impact of diet on the gut microbiome, we made the decision, well, let's choose cohorts that differ based on their diet, and to the extent possible, the most extreme opposite diets that we can select from. And we chose to go with vegetarians and omnivores, just with the basic understanding that you would expect to see different microbiome profiles from these 2 groups of diet cohorts. So we chose multiple donors per cohort. Some of these donors gave multiple bowel movements, and we collected a lot of the fecal material. We collected bowel movements from these donors, and we pooled it. We put all of the fecal samples from any one cohort into a pooled batch, and we homogenized it. This is a Ninja blender of a smoothie that I got off of a Google image search. This doesn't happen to be a picture of the actual fecal material being homogenized, but it's just for illustrative purposes. But we pooled and homogenized this material, and then we made 5,000 aliquots of the material from each donor cohort. So it's a total of 10,000 aliquots, but it's 5,000 aliquots per cohort for a total of 10,000 aliquots. Each aliquot has about 1 ml of material in it, which represents about 100 milligrams of the actual fecal material. We put water in it to make it more amenable to homogenization, etc., to pipetting. And importantly, as I mentioned, we put water in it. We did not put anything in it other than ultra pure water. It is unadulterated, neat material. And I'll just preface that by saying, we did try during our R&D phase putting some of these proprietary storage buffers, these stabilization shipping buffers in it, and one way or another they created problems. Many of them did exactly what they were supposed to do, what they're advertised to do, like preserving the nucleic acid. But that might severely interfere with like metabolomic measurements, which we did see. These storage buffers have high concentrations of salts and surfactants and detergents and various things, and these can and do interfere with some of our measurements. So we wanted it to be unadulterated, and we were able to do that. So this is just pure fecal material and water. Okay, so this is the first part of this is the manufacturing and making these thousands of aliquots, but now comes the real hard work, and that's the exhaustive characterization. So now we have thousands of aliquots of our vegetarian and omnivore material, and we did multi-omic characterization on it. We did, I'm just going to go through this pretty quickly, but we did 600 megahertz NMR metabolomic measurements. We did 700 megahertz NMR metabolomic measurements. We did liquid chromatography, mass spec metabolomic measurements. We did flow cytometry measurements. The various microbial taxa that exist in fecal material represent different species. There's fungi, there's bacteria, there's archaea, and these have different cell morphologies. And in some cases, these different cell morphologies were differentiated or are apparent on flow cytometry. So it was a way of assessing some of the homogeneity and stability properties of it. Of course, we did deep shotgun metagenomic analysis. This is the NGS approach that tells you taxonomically what's there. We did culturomics working with a third-party company. We were able to isolate about 2,000 isolates from this fecal material that we now have in our freezer. So you can imagine that, in the future, we could build up those 2,000 isolates from the bottom up to produce sort of this engineered fecal derived material. We also did moisture content. Okay. Sandra De Silva, my colleague, is going to talk to you about the metabolomic measurements in the second half of this talk, and I'm seeing I'm coming up to my time. So let me just mention that the original report of analysis that we came out with includes these characterization methodologies that are shown here, but we're continuing to do more exhaustive characterization. We have a scientist here now doing bioinformatic analysis of the metagenomic data to develop metagenomic assembled genomes, also known as MAGs. We're spinning up some metatranscriptomics or gene expression profiling from these materials, metaproteomics, lipidomics, and virome. So you probably know that the NIH is launching the Human Virome Project, and they're clamoring for the need for standards, and we're hoping that this fecal material will be adopted by the virome community as a human virome standard. We imparted some ground truth into this material while we were manufacturing it. We grew up 3 pure isolates of bacteria that are environmental bugs that should never ever be found in human fecal material. So some of these were like extremophiles. But the point is that we grew them up to a specific cell enumeration, and we spiked them into the fecal material at about 1% each. So 97% of that signal should be from the native fecal material, but about 3% of it should be these spiked in organisms that we introduced that provide sort of a normalization effect. And I'm just going to show you very quickly. Here are 5 bacterial species who were put through sort of a log dilution series, and you can see the slopes of the lines are supposed to be linear according to a dilution series. This is the data without considering the spiked in organisms, and you can see that that same data, when normalizing to the spiked in organism background, the response function, the linearity becomes much better. This is sort of analytical chemistry 101. You introduce an internal standard, and you can improve your analysis and your analytical method. Okay, this material is available to the public. It was released on March 17th. It was about 6 years of hard work. And I'm going to turn this over now to my colleague Sandra De Silva, who is going to tell you more about the metabolomic analysis that was done with this material and other versions of this material. So Sandra and I are going to kind of tag team this. Sandra, you can take it away and say click when you're ready for me to advance the slide.
All right, thank you, Scott. All right, so click. Okay, so to set the stage, to share with you all the different steps that we went through to develop this reference material, I would like to highlight this recent publication that truly state the need for reference material that we see reported in the literature, right? So in this article, it states that there is no established ground truth for what molecules or species to expect in a stool. So there are tons of molecules, tons of microbes, but we really don't know what exactly we are looking for. And ideally, we should all have the same type of results, but in reality, this doesn't happen, and this has been a challenge for the metabolomics community. So it's clearly there is a need to elucidate unknown metabolites, address the quantification, data reproducibility, and comparability. Click, click. And part of this challenge, it's really related with the complexity of stool samples, right? There is the biological variance that it's related with diet, age, the microbiome composition, the microbial metabolism. In addition to this, there is this huge chemical diversity, diversity structures, stability of some of the chemicals property, and in the end, the method variability, right? The analytical tools that are usually used to analyze those samples also bring their own challenges in order to keep the reproducibility of the material. So based on that, so a homogeneous stable material, like for instance, the RM8048, so clearly could help to set up that stage and bring some reproducibility and comparability for these studies. Click. So, and also it provides a matrix match material that is similar to the biological complex of the samples that are being targeted in the studies, and also helps to harmonize measurement across different studies. Click. So I will start talking about how we started this effort. This was our first study when we had the opportunity to look at some preservation method to preserve these two samples. And as you can see here in the graph, I hope you can see my cursor. So we test 5 different types of preservation method, including these 2 ones highlighted in red. They are preservation method used to preserve samples for metagenomics. And they were usually fine because they were targeting only the DNA. It didn't matter what happened with the cells or with the metabolites in those samples. But however, we were looking for a preservation method that would be applicable at the same time for metagenomics and metabolomics. So in this preservation method, so on the graph on the left side, you can see the results for metagenomics, and on the other side, you can see the results for NMR. What I like about this result, it's the 2 samples at 4 degrees are the 2 samples far away from the other ones. That was really interesting to see. Indicating that storing the samples at 4 degrees, so it can change the microbial composition and also can change the metabolite composition as well. And another thing that I would like to emphasize is that the DNA RNA shield and omenogene gut, so it really it was two preservation method that we could not even think about it, because it completely stopped the LC mass spec. So our colleague that was running those samples had to spend the entire week after adding those two samples there, cleaning up her system, because the preservation method really didn't work for her instrumentation. So those results, in the end, we realized that lyophilization and water at -80 were the two storage conditions that we could move forward. And also, as I just explaining, the some of the preservation method not comparable with metabolomics instrument. Click. So with that result, we had the opportunity to present at a workshop held in collaboration by NIST and the IFANS. And the idea of this workshop was to bring the metabolomics community to discuss which type of metabolites they would like to see in this potential reference material, because by that point we were still in discussions, and which also type of storage condition, and also the format for this preservation that they would like to see. From the output from this workshop, we there was a consensus for the need of a whole stool reference material, and the community believed that would be a material that would improve the measurement reproducibility and also brings data comparability amongst different measurement capability. Next slide, please. So from this workshop, we had the opportunity to register the result in a publication. And in this publication, we had highlight all the outcomes from the workshop, including the list of metabolites that the community would like to see characterized. So that was very important for us, because we use this as a guide to really characterize our material in the best way that would be useful for the community. Click. So in addition to the workshop and the pilot study, we had the opportunity to run a metabolomics inter-laboratory study. And in that stage, based on the first, the pilot study, we were able to have a pilot batch that it was manufactured for us to optimize protocols, to see what were the techniques and also methods that we would use to characterize our material. And also we were able to use this material to get the directly output from the community. And the idea of this interlab study was to evaluate the fitness for purpose and also to support the assurance of metabolomics measurement. And in addition, to assess the state of the field with respect to the data comparability. It was truly basically giving to the end users and ask them what they like, what they didn't like it, and how we could improve the material, and also see the data comparability. So basically, we had two samples, Viga and Omenovore. They were in aqueous solution and also lyophilized. So the samples were analyzed by three different techniques: NMR, mass spec, and GC. So once the participants in this study had the results, they sent the results to us, and then we analyzed. We also were able to register that study in a publication that was released last November. Click. A little bit more about this study. So we had the participants, mostly from US, also from Europe and Canada. So the participants, they came from academia, government, and industry. We had 26 enrolled participants, in which 18 of them returned data to us. And the participants, they were allowed to use the analytical techniques that they were more comfortable with. We didn't set up any condition for that. And we just asked for them to send to us all the annotated metabolites, anything that they could detect, and also the top 20 metabolites ranked by abundance, which was, the abundance was signal intensity, peak height, peak area, concentration. And in addition, we had some of the participants that they use more than one techniques to analyze the samples, and you can see in the table on this side. Click. All right, so just to give you a taste of this result, here we have a pie chart for the three techniques, highlighting, for instance, LC mass spec, that the participants, they produced 25 data sets, and that came from 14 participants. And this happened because some of the participants, they use different methods to analyze their samples. And a total over 1,400 metabolites were reported. And the interesting part is, once those metabolites were standardized by RefMet, so we came down to 8,510 unique metabolites. And in this, for LC mass spec, only 36% of the metabolites were reported more than once. And on this table on the side of the pie chart, so indicates the frequency that the metabolites were reported. And for instance, 4 metabolites were reported across, 4 unique metabolites were reported across 14 participants, while 0 metabolites were reported across all the participants in that study. If you go for GC mass spec, so 6 data set was reported from 6 participants. So a total of 875 metabolites were reported, and it turned out to be 544 unique metabolites, and 58% of the metabolites were reported more than once. In the case of NMR, we had a total of 5 participants reported, all 5 participants reported NMR data, and that 246 metabolites were generated after RefMet normalization. And 9 metabolites, for instance, were reported across all 5 laboratories. Click.
Sandra, you have like 4 minutes left.
Yeah, I am tagging here. It's okay. For me 6. Okay, thank you. Click.
Okay, sorry, I thought I did. Go ahead.
Okay, yeah. So this was just for the top 20 metabolites, where you have the information before and after RefMet. So you see an increase in multiple metabolites report after RefMet, indicating the importance of standardization, as we all know about that. Click. Okay, the lessons learned here, you can click a few times, Scott. Yes, lessons learned from this study. It's the material was amenable to multiple metabolomics extraction method analytical techniques. The two diets present unique profile, as was also demonstrated in the literature by the participants who published their analysis. They did the analysis for us, but they also published. And the reference material, having these two reference materials, it's very valuable because of their ability to really look at the different workflow and distinguish between different population. We opted to move further in producing the reference material in ACOS format because that was close to a real sample, and also because we want to minimize any loss in the short chain fatty acid. And this study compile a very comprehensive list of metabolites because of the different analytical platforms that were used, and also methods to analyze the sample. Click. So here you can click again. So here I am going to show the two main platforms that were used to analyze the stool material, and it's part of our certificate. Click. So this is just the protocol used for the NMR samples. We use the polar metabolite extract, and also we use another method that is commonly used by the community, where we look only on the water-soluble metabolites. Click. This is just how it looks like NMR spectra. So you cannot get much information just looking to this. Click. But once you put this in a PCA plot, you can see that these two samples are very different. And this is what we were looking for. Click. So to give a metric for the homogeneity of this material, we use the coefficient of disagreement. Basically, we were looking for the potential disagreement between any pair of samples, and with that, we could come up with a percentage. So if you are more interested to learn more about that, our statistician can talk more about it. But for the omnivore samples, our COD value was 10.2%, and for vegetarian was 6.8%. For this type of sample, because of the qualitative application, we deemed that it was fit for purpose. Next slide, please. So we also look at the stability of the samples, where we could see some changes in a few metabolites over time. So again, because this is a qualitative reference material, the material is still applicable. And what we are planning to do is to continue monitor this stability and see how it goes, and then maybe redefine the certificate again. Next slide, please. So for the LC mass spec, so this is just a summary of the two different types of analysis that done, was on target and the target. And we look at the positive and negative ionization mode. Click. So again, for the homogeneity for LC mass spec, you can see for the positive ionization, negative ionization, that you see the two different samples as we were looking for. If you look at the variance, the relative standard deviation, it's below 20% across all the different samples, including the two different techniques that were used. Next slide, please. So, so we had two different instrumentation, two different types of ionization, and three different types of algorithm to really set the annotation and brings confidence to the annotation of metabolites in the sample. Next, please. You can click again. Okay, so going across all these three different algorithms to annotate the metabolites and be confident, in the end, we set for omnivore diet that 107 metabolites were annotating using mass spec, while 121 was annotated for vegetarian diet. In the case of NMR, we have 81 metabolites for vegetarian and omnivore each. And also some compounds show change over time, but the material remains applicable as a qualitative reference material. Click. All right, and just to finalize, so the stool material brings you reproducibility, right? So in order to add to this effort, we also came up with a fecal calibrant solution. This is the RGTM 10212. And the idea of this solution, it's a material with 70 different metabolites where we know which metabolite it is, and also we have quantified the material by NMR and mass spec. So because it's RGTM, so we developed this material to give it to the users to use and provide us some input. So the material is available. If you click in this barcode, the QR code, you can go to the web page, and the material is free of charge. And we would love that the users test the material and then bring us their feedback, because our intention is to really transform this in a reference material, and we would like to do this in a way that the material will be useful for the community. I think this is my last slide, and I'm going to transfer to Stephanie. Thank you.
Thanks, Sandra. So Stephanie is going to conclude with just some thoughts on our ideas around data sharing, and then we will go into the Q&A.
Thanks, Scott. So I'm not going to take up a lot of time with this, but I think we would be remiss if we didn't try to create a really rich data source from this reference material and what we're going to be able to collect. So the model that we're currently working with to leverage things that we have up and ready to go at NIST and will be easiest to implement is that there will be a link available to those, and this isn't up there quite yet, but hopefully shortly it will be available through the shop page. And when you purchase this material, it will be a link to request access to a box or something similar, cloud-based data storage that will be behind the NIST firewall. We'll send you upload instructions. The lab will provide the metadata, so how when we'll have an Excel or some sort of CSV that you can fill out with all the files about how you process the sample, as well as your raw files. And we might have a formatted ID table, so if you have either metabolites or taxonomies, maybe you might be able to upload those as well. Every 3 months or so, NIST will do a data pull, so we'll grab all the raw files. Labs will receive a unique ID, so when we go ahead and share this data back, we'll tell you your unique ID so you can see how it fits in, but everyone else would just see you as lab 4 or something like that. This de-identified data will go up on our data.nist.gov or MIDAS portal so that you can have access to all of the raw files as well as the process data as well. And then we'll have an interactive way to look at that data and just see, okay, here's my lab's results. How does that compare with other folks? And so just as an example from the metadata, click. This is what we would hope that this would look like. So you could have some sort of PCOA, ordination plot. You could hover over the dot and say, here's the lab ID, here's the kit that they use, the extraction type, those types of things, and see how changing your methods might lead to, a difference in the data that you generate. So this is our grand vision, and hopefully we'll have it implemented within the month. That way, when people purchase it, they can go ahead, sign up. And this would just be for the data generated from RM8048, whether that's the vegan or the or vegetarian or omnivore samples. It wouldn't need to be all of the data that you're collecting, just a very limited profile. And with that, I will turn it back over to the organizers, and Scott and Sandra, if there's any questions.
Thanks, Steph. Let me just take a final moment to acknowledge what has become known as the POOP team here at NIST for obvious reasons. This is the folks that developed this fecal standard that you just heard about. So a very talented multidisciplinary team. And I also want to acknowledge IFANS. IFANS provided some financial support for this, and where IFANS financial support sort of manifests itself is that we sell this material for a fixed price. And through the support that IFANS gave us, that price that we sell it for was reduced probably about 20%. So we're able to reduce the cost of the cost recovery model, and to get this material into the hands of the end users, the stakeholders, in a little more affordable way, thanks to the financial support that IFANS provided. So we appreciate that. And now we're done.